eukaryotic cells, right? Eukaryotic cells such as your immune cells that are inside you. That means we're looking at endocytosis. There's 3 types of endocytosis that we talked about, right? First one we talked about was pinocytosis or penocytosis, however you want to pronounce it. Second one was receptor-mediated endocytosis. Receptor-mediated. Receptor-mediated. The receptors mediate the entrance of the ligand into the cell. That's like you go into Walmart, and there's a greeter there, and if it's a good greeter, he greets you, asks you what do you want. You say, I want to find yogurt. So he will grab you by your shirt and drag you over to the section that is where the yogurt is at, right? Receptor mediated, okay? The other one is phagocytosis. Morning. Did you think it was 11 o'clock too?
No. I thought it was 11 o'clock, but I didn't know that it was 10. I thought it was 11.
Okay, so pinocytosis, receptor-mediated endocytosis, and phagocytosis. Now, the characteristics of pinocytosis is basically the cell invaginates and forms a endosome. Inside that endosome is a liquid sampling of its outside, right? This is something that occurs like in your digestive system, where you get pinocytosis inside your intestines, right? So it takes a liquid sample of what's out there. That endosome, which is a vesicle, is now inside that cell, right? It's an interior of the cell. You have another vessel that's called a lysosome. Lysosome contains enzymes. And we know what enzymes do, right? What do enzymes do? So these break things down. So you get a fusion of that endosome with the lysosome. The contents of that lysosome will enter inside there and start breaking down whatever stuff is in there, right? So kind of like digesting in your stomach, basically. The lysosome is the stomach of the cell. So it breaks that stuff down. Once it breaks it down, any waste that is comes from that needs to get outside the cell, right? And so that endolysosome fusion right there, that one vesicle now will move to the other side of the cell and then be released through exocytosis. Okay, so you have endocytosis, the entrance, you have the processing of it on the inside, and you have exocytosis. This whole process is called transcytosis, meaning transitioning through the cell. Transcytosis is the whole process. So that is pinocytosis. So the basic understanding here is you have 3 different types, right? Once that vesicle enters into the cell, the same thing is going to happen to it no matter what, okay? The only difference is how it gets in. How it gets in with pinocytosis, you have invagination of the water or liquid portion out there, brings it in. Receptor-mediated endocytosis, you have specific binding of a ligand, L-I-G-A-N, specific binding of a ligand to a receptor. Once that binding happens, that causes the cell membrane to invaginate, just like it did with pinocytosis. When it invaginates, it's going to form an endosome, right? That
What did you say at the beginning?
receptor mediated, specific binding of a ligand.
Of what?
Ligand, L-I-G-A-N. Okay, specifically binds to it. That causes the cell membrane to invaginate. Once it invaginates, it's going to form an endosome, just like it did with pinocytosis. What's going to happen to that endosome next? Same thing that happened to the endosome in pinocytosis, right? You have a lysosome that binds onto it, fuses it. The enzymes from the lysosome digest that content that's in there, and the waste is put out through exocytosis. Right? Why are you looking at me like that? You have a question on it?
No, I didn't get it.
You got it? You didn't get it? Okay. What part did you not get?
No, it's because I kind of came through the middle. No, it's because I came through the middle of it, so I didn't get it.
Bless you.
Okay. We're talking about how things get into the cell, endocytosis, right? There's 3 types. Pinocytosis, taking a water sample.
Okay,
Coffee.
the essay question reads, describe the process of endocytosis, give examples of offered types.
Yes.
On our essay question, if we describe the endocytosis occurring like when the cytoplasmic membrane surrounds a substance and brings it into the cell, right? And then we can describe the phagocytosis, like bringing a solid into the cell, pinocytosis, bringing a liquid into the cell, exocytosis, reverse of endocytosis, and it exports substance from the cell. So does that cover all of them? Good enough.
Okay, so,
Essay.
let's see.
Or, and then we have to put the receptor mediated.
Endocytosis is the act of a cell bringing act. It is active, it is live, it is doing something. Act of a cell bringing a substance into the cell by 3 methods. First, pinocytosis is where a cell takes a liquid sample of its environment and brings it into the cell by way of invagination. Can't spell invagination. The endosome that is formed fuses with a lysosome where the enzymes contained within the lysosome digest the contents of the endosome. Okay. The waste is exported from the cell through exocytosis. Okay, right? Now, I've already described that first thing, right? Now, if I want to talk about the other two, I could say receptor-mediated endocytosis is where a specific binding of a ligand causes the invagination of the cell membrane, bringing the ligand into the cell, right? We're at that point. Now, the receptor-mediated binds onto the receptors bind onto the ligand, bring in that substance through invagination, right? Forming the endosome. Okay. Now, I don't have to explain all the rest of it because I can therefore refer to what I said before. And I can say that the processing of this substance is similar or the same as the processing of the substance in pinocytosis. Done. Move on to the next one. Right? I don't have to write all that stuff out again.
So we just write like what we wrote for pinocytosis, and then we said like specific binding of ligand happened, and then
Yeah.
after that, it's similar to what happened in pinocytosis.
Exactly.
Okay.
So you don't have to write it all out again. Okay? Then we go to phagocytosis. Phagocytosis involves the use of pseudopods by a cell
What's the use of that?
pseudopods. Pseudopods. I don't know how to spell pods, I'm just lazy. P-O-D-S.
Is
P-S-E-U-D-O. Pseudo, as in false.
Wasn't pseudopods
Pseudopods.
used for every stage to bring the liquids, the solids? No?
No, only in phagocytosis.
Okay.
Because different cells do different ways of bringing it in. So, now I can't go down that road. Anyways. Well, I was gonna start talking about MHC cells and your immunity and your specific immunity.
Don't get us so don't be here.
Dr. Chung would. But anyways, pseudopods reach out and grab, for example, a bacteria. Just use that as an example, and bring it into the cell. The only difference at this point is that vesicle that has been formed through that is called a phagosome. But that phagosome goes through the same process as what happened in pinocytosis, right? So as long as you refer back to pinocytosis, the same thing happens once that vesicle is inside the cell, except for the vesicle is called a phagosome, not an endosome. Scientists like to do that to confuse you.
So it goes through the same process?
It goes through the same process.
And then
So-
except
And same process all the way through to